Hello everyone, welcome back to my channel once again. And I would like to thank you all for your kind support and motivation towards me and towards my channel. So please guys keep loving and supporting me like you're doing now. Today I'm going to present about the cloning. What is cloning? Cloning in general term that describes a number of different processes that can be utilized to produce genetically identical copies of a biological entity such as animal, either plant or any prokaryotic cells or any eukaryotic cells. So during the cloning process, it makes any individual with identical or virtual identical DNA. So it either can happen naturally or artificial in laboratory. In natural way, like we can give an example of twins, identical twins deliver from any animal or human, and artificially we can make replicant of any bacteria or virus in laboratory. And the dolly sheep is first ever cloned animal in history. Now I'm going to give you the types of cloning. I'm gonna give you the general perspective of three different types of cloning that we usually use in biotechnology. First is gene cloning, second one is somatic or reproductive cloning, and third one is therapeutic cloning. And in biotechnology, we do gene cloning most often, and in some cases, in some laboratories, biotechnology deals with somatic or reproductive cloning and therapeutic cloning as well. However, in plant genetics and or most of the biotechnological experiments, they generally go with gene cloning. And I'm talking from the plant or agricultural genetics perspective, so I'm going to stick more with gene cloning, but I'm going to give you a general idea about what are these three cloning types. Now, first, I would like to go with gene cloning. What is gene cloning? It is making of exact copy of particular DNA. The particular DNA that encodes for certain gene type. So, generally, gene cloning is used to known by either gene cloning or sometimes DNA cloning or sometimes molecular cloning or sometimes recombinant cloning as well. And cloning use the approach of recombinant DNA technology. And how do you obtain the gene for gene cloning? In our cell, within the nucleus, we have chromosome, and chromosome have DNA, and certain sequence of DNA encodes for certain gene. For example, if somebody else is like highly dwarf, and we can identify the gene that is causing dwarfism. So in this way, the certain sequence in his DNA is representing his dwarfness. So to identify like this section of DNA, we have to do this certain analysis. Then after identifying the gene, we use this certain DNA sequence that encode that gene, and we take that gene from the original organism such as from plant or animal. Then we put that sequence or that fragment of DNA into the recipient vector DNA, and we recombine them, we transform into this certain host cell, and we grow them, and in this way we clone the certain gene. Gene cloning in agriculture or in plant helpful for making mutation or making genetically modified organisms or genetically modified plants. And in some cases, it helps to genotype the plant, certain type of plant, and to express certain type of beneficial protein in the plant as well. Therefore, in agriculture, I can see the perspective or scope of gene cloning into making genetically modified plants that can have the high yield attributing quality, or some agricultural plants or animals like can be better in giving production. Next one is somatic cloning. This cloning is called as a reproductive cloning in some cases, therapeutic cloning in some cases, and it uses the nuclear transfer method, and it creates ovum with donor nucleus. And what happened in somatic cloning is it uses egg cell and somatic body cell. The somatic body cell with desired genes, and we already knew that nucleus is a storehouse of DNA and a specific gene. That DNA will encode some genes, and if you want to clone that gene into the next generation, what we have to do, we have to fuse that somatic nucleus into the denucleated egg cell. The egg cell should have the removal of nucleus, and we can fuse this nucleus into the egg cell, and we can grow into the certain medium, and we can replicate them. The egg cell carrying specific desired gene from the somatic cell. So yeah, if you like, if you want to take an example of human, like if somebody else is infertile and they want to give birth, what we can use a similar kind of cloning procedure. Like we can get egg cell from certain woman, and we can fuse the nucleus from somatic body cell of male, and they can give birth of like any child having the specific desired genes. However, we cannot perform such kind of cloning procedure in human. So we can do this kind of procedure into the animal. We are allowed to do with animals because we can use it for the better production perspective, or making like for the disease free or some immune perspective cloning in animal, but we are not allowed to do in human. So similar kind of procedure we can do, we can replicate in man, like other mammals such as cattle, to improve them for the better yielding properties. So the concept is we can use the any body cell, and we can fuse into the egg cell, and we can replicate them, and that replicant would have the egg cell from same or different organism, and somatic cell from same or different organism, and in some cases this somatic cloning reflect into the therapeutic cloning, such as tissue culture. And the next one is therapeutic cloning. Therapeutic cloning is the process to make embryonic stem cells, and that embryonic stem cells should have the same DNA as donor cells. And this method of cloning is used to understand the disease and to develop the treatment towards that disease. And this cloning usually used to call as a stem cell transplant. And I have already mentioned that the somatic cloning sometimes reflect into the therapeutic cloning. So sometimes the therapeutic cloning used as another phrase for somatic cloning. Anyway, how therapeutic cloning been done? And I am here giving you an example from developing insulin producing pancreatic cells from type 1 diabetic patients. So what how do we carry out this cloning is we take an oocytes, unfertilized oocytes, and we have to remove DNA from there. And from the type 1 diabetic patients, from his or her skin, we use the specific DNA into that oocytes. And after transferring into the oocytes, the blastocyst happened, and it will give the embryonic stem cells lines. So these embryonic stem cells line will be later on differentiated into insulin producing pancreatic cells. In this way, the insulin producing pancreatic cells will be developed, and it will later on used for the insulin production and treating the diabetes. And this much for today. And taking the perspective from agriculture, I am going to talk much about the gene cloning procedure and everything that includes into the gene cloning, such as cloning vectors and the procedure and methods in next video. Till then, please keep watching my videos, keep supporting me, keep motivating me, and please do not forget to leave your suggestion, feedback, and comment on the comment section below. Have a very good day. Enjoy your day.